Hello everybody. So my name is Pratyaksh Rastogi. Hi guys, how are you all? Okay, I can see a hi from Priyanshu. Hi Priyanshu. Hi Shibrin. I'm sorry if I'm pronouncing the name incorrectly. Hello to you. Okay, I can see there is one viewer already joined in into the session, so I welcome you. Hello. So this is Pratyaksh Rastogi guys, and I welcome you all to my online webinar. And I'm a biology teacher at Academy, and I teach both, that is zoology and botany. By profession, I'm a physical therapist and I'm a teacher by choice. I love to teach students, and I love because I'm so much fascinated with biology. Okay. So I welcome you all. So beginning the session with, first of all, just wanted to make sure, is my audio, my video is absolutely clear to you? Are you facing any issue in hearing my voice or watching my video? So just let me know. Okay. You can just drop a message simply in the chat box so that, let's make this session a little bit interactive so that I will also get enjoyed, you guys will also get enjoyed, and we'll be good to go. Okay. So second question I just wanted to ask, how your studies are going on. Are you on track with your studies? Okay, because right now guys, the month of February is already going to be introduced just day after tomorrow. It means that you only will left with 3 or 4 months more, right? So you should at least be able to complete 85 to 90% of your syllabus. So is that so? Is everything going good, okay, with your studies? If not guys, then I would like to tell you that, stay more focused into that. Just make some dedication, okay. Make some resolution that yes, you have to complete your syllabus first. Hi, hello. Okay. So, just what you have to do is, first of all, go through the NCERT. Just keep NCERT on your fingertips. That's the most important part, guys. Okay. Especially when it comes to biology, you all would understand and agree with me that, it's really important for you. Then what we have is, okay, so yeah, so you should, that's what I said, that you should be completed with at least 85 to 90% of your syllabus. Okay. And yes, just stay more focused and you will be good to go. Just be relaxed. Okay. Watch the videos whatever we are preparing for you. Okay. And yes, you just attend the test, the mock test which are giving to you. Just go through all the additional material what is being provided, and yes, everything will be fine. Okay, don't worry. Okay, so guys, this is my introduction. You can all go through that. So that's what I told. My name is Pratyaksh, and I've been associated with Academy, for a few months now. And right now, guys, what I'm doing is I'm doing some researches about, soft tissues participation in sports science. Soft tissues include your ligaments, your tendons. I hope you know all of that, right? What is a ligament? Ligament is something which connects two bones together, right? And what is tendon? Which connects muscle to bone. So ligaments, tendons, muscles, your nerves, your vessels, everything, it's included into soft tissues only. Okay, everything which is surrounding the joint is called a soft tissue. Okay, so I'm doing some researches over there, into the sports science. So guys, now let me introduce you all to the Academy. So what is Academy? We actually organize the classes in the similar way for you all. Apart from that, we also have the other facilities, and where you also have the benefits. What are those is, first of all, we organize the live webinars like this only on our YouTube channel. So you all would understand that YouTube is very accessible these days. You should have no problem with accessing the YouTube. What you have to do is just create a Gmail ID and you are good to go, right? So that's why we chose YouTube. We are posting our videos in YouTube so that you can watch our videos at any time. Okay. Apart from that, we are providing you study materials. In that study material, we try all the faculties try their level best to provide the material in a concise way, and we tend to provide the precise material to you. We add additional matter also wherever it is needed. For example, in some topics, even if I would talk about biology itself, in some topics there is additional information needed, so you all will be able to access that information through our study materials, right? Apart from that, what we do is we organize, or we take some test of you also, like it's a kind of a like flipped classroom method. You all would agree with, you all, if you all know what is a flipped classroom method, let me tell you. In flipped classroom method, what happens is, first of all, we provide you the study material in the form of PDFs. You just go through that, you just take a view of that, okay? And then after you get some idea, we provide you a webinar. In that webinar, since it's a video, so you are able to better see everything, you are able to better capture everything what is being what was being written into the PDF and what is being explained to you on the board, right? So and after that, once you get everything, then we take a test for you. So in that test, you are also able to self evaluate yourself wherever you are lacking, which topic or which concept you need to be more focused into, so that the whole chapter will be into your hand. Okay, so this is what we do in our flipped classroom method. And let me tell you one thing also, even the most precious institutions or organizations in our in India itself, for example, if I name IIT Delhi or NIITs, they all are adapting the same teaching method, right? Then the last thing what we do is we also check your homework. So we do have the mentors available over here. Those mentors are responsible to inform you, like you have gotten this much marks, and which question you did mistake in, and so that they can guide you accordingly. Okay, then proceeding to that. Okay, so now additionally to that, what we have is, now guys, you can do one thing. We also have a website for Altademy. Then the website is altademy.online. You can all do one thing, just visit the website, go through all the benefits and the features listed over there. Let me give you some brief idea over here. So guys, we have 3 plans. Okay, the first plan is light plan, second will be smart, and third is proficient. In light plan, what we provide to you all is, first is the interactive live classes. Right now I'm taking your class, you are able to listen, and it's a kind of like because video, a student is able to better capture or grasp the things in a video. Okay, so that's why we organize the classes over the YouTube, so that you can watch us, you can watch our content, and you can easily understand the matter. Then in the second thing, what we have is structured course and PDF. That's what I told. So we provide you the PDFs, okay, for every topic what we cover. Then we have live test. I told you already that it's a part of a flipped classroom method only. So we also organize the test for you, where you are better evaluate yourself, like where are you lacking or where are you more improved. Okay, then at last we provide you the physical notes as well. And then when it comes to smart plan, so we have all the benefits what you are getting in light plan. Additionally, into the smart plan, what you are getting is one-on-one mentorship. So guys, I told you we have mentors available for NEET and for JEE students separately. Mentors are available for you all 24/7. You can ask your doubts if you are having any problem with the syllabus, your syllabus is not on track, you need help in managing the timetable, you need some schedule. What you have to do, just reach out to those mentors and they will help you in every way. Also guys, we have a Telegram channel and a WhatsApp group also. So once you will get interested, you will be joined through that, and you will be able to see the mentors activated over there. Okay, then we have live doubt solving. So guys, in live doubt solving classes, what we do is, for example, if you are solving any topic, if you are solving previous year question papers and you are getting any doubt in there, you are stuck. Okay, what you have to do is, we will once the syllabus will be completed, what we will do, we will organize these kind of classes for you, where you will just be able to post your questions either on our YouTube channel, or you can also reach to us through our Telegram channel. And once we see your questions, we'll solve that out for you. Like every day many students just ask us the questions, like what do we have to do in this, especially when it comes to physics and chemistry, because it contains numericals. So students are more often stuck with the numericals, with the mathematical portion. So yes, you will be getting the live doubt solving sessions also, whereas we will also take mock test for you based on the current NEET pattern, because the NEET pattern will also get changed this year, right? So we will organize, we will prepare some test for you based on that pattern only, where you will be practicing yourself about the pattern also, because practicing the pattern is also important in order to not waste the time in the exam, right? Then when it comes to proficient, so proficient contains all the benefits of smart plan. Additionally, what we have is, we have an additional special course for you in which we will provide you, an additional information. For example, some students are preparing for NEET only, but there are students who are preparing for NEET, AIMS, and JIPMER also. So for AIMS and JIPMER, you need some additional knowledge. You need to be you need to be on an extra edge, right? So that's what we provide to you. Whatever additional information you will need, we will provide to you. Also, guys, additionally to that, on our Telegram channel, on our WhatsApp group, you will be getting all the updates related to your NEET exam, related to the live classes what are being organized for you all. So, just visit the website, go through all the plans, and just choose whatever plan is suitable and beneficial for you. Make up your mind accordingly, and you are good to go. Okay. Then, guys, I would like to. Okay, so starting with the topic. So for today, I've selected the topic again, which is from the chapter Molecular Basis of Inheritance. It is a chapter from your class 12th. Okay, so I have chosen a topic. First of all, I will talk about how DNA was confirmed as the genetic material, and second thing, we will talk about the DNA replication, how replication goes on. Now, what is a replication? Replication is a process in which there is another copies, which is made for the original copy. That is called a replication. Replication is nothing but copying that. Okay, just say when you go to a photostat shop, to have a photostat, it means what you are doing is you are creating a replica of that copy. You are creating a replica of that page. Okay, so similarly in DNA there is a replication. So guys, starting with, we have biochemical characterization of transforming principle. So I told you what was transforming principle. I explained it to you earlier. In transforming principle, it was just a test which was performed by Griffith. And in that test, what he did is he selected the bacteria, and what he did is he found that the bacteria was evolved, and there was two types of you can say the stains or the strains of the bacteria which were produced. The first was S strain bacteria and second was rough strain bacteria. So S strain was smooth because of the mucopolysaccharide mucopolysaccharide coat over there, whereas the rough bacteria did not have any. So now what happened? Now Griffith, what he did is he took a mice as a test subject, and he infected that mice with that S strain bacteria as well as the rough strain bacteria. So what he found is the mice which was infected with S strain, they got died because of the because of pneumonia. Okay, whereas when you talk about the mice which was infected with R strain bacteria, it did not have any symptom. It was inactive in the body of the mice itself. So what he found is that the S strain had something which was able to infect the mice physiological function. What was that? That was the genetic material, but he was not still able to recognize whether it was a protein or it was a DNA, right? So, and after that, what he did is he took S strain, he heat killed the S strain. Basically, in the presence of heat, the mucopolysaccharide got removed. So he heat killed the S strain bacteria, and then he combined that S strain with the R strain and infected into the mice. Now what happened? That you can say that the mice was alive. Okay, but okay, so this was the transforming principle. What was what was performed by Griffith. Now after that, what he found is that the S strain was the ultimate. You can say S strain had some genetic material which was also converting the R strain into a into killing the mice. Okay. So similarly, guys, what was the result is now we have a biochemical characterization. Now, how was it confirmed? What was that genetic material? Was that the protein or was that the DNA? So you can see that prior to the work of Oswald Avery, Colin MacLeod, and Macklin McCarthy, the genetic material was thought to be a protein. Okay, but what is a protein? No, it was a DNA. Okay, so what they do is they took over the Griffiths experiment. Okay, they took those S strain and R strain bacteria. So guys, before proceeding ahead, I would like to again tell you that this is the link for the Telegram channel. This is the QR code. You can just take out your mobile and you can scan this QR code and join through this Telegram channel. You can see the ID for the channel is alt NEET, and this is the name of the channel, Altered ME NEET. Okay, so you can see we already are posting these kind of important information for you, like PDF material, free classes, announcements, discounts, and prizes for you all. Okay, so just in order to be updated at every time, you have to join this channel. So what these Avery, McLeod, and McCarthy did is they purified biochemicals which were present over there. So they took proteins, DNA, RNA, etc. Now what they did is they basically took these biomolecules from the S heat killed bacteria, right? So to see which one could transform R cells into S cells. So that's what I told you. Like what they did is when when Griffith took S cell along with the R cell, so what he found that the mice was dead because S cell converted, heat killed S cell converted R cell into the S cell only. They were able to copy the machinery of the R cell itself. So the mice was ultimately dead. Okay, so basically they all three basically extracted the biomolecules from the S cell bacteria, and then what they discovered is that the DNA alone from the S bacteria caused R bacteria to become transformed. Okay, once you will perform test, you will be able to know that yes, it's a DNA which is actually transforming the you can say the R bacteria into the S strain bacteria. Okay, and S strain bacteria was fatal. So guys, this is the YouTube channel what we have. The name of the channel is Altered ME NEET. You can just search it over here. What you can do is just subscribe it and hit on the bell icon so that whatever videos we will keep posting for you all, you will get a notification over your mobile, over your YouTube. Okay, so that you will not miss any class for your physics, for your chemistry, and for your biology also. Okay, next what we have is they also discovered that protein digesting enzymes, proteases. So guys, I told you before as well, whenever at the end of the enzyme, whenever at the end of the name it comes ase, it means it's a digestive enzyme. Okay, digestive enzyme means that it degrades that biomolecule. For example, protease means they are degrading, they are digesting proteins. Okay, so as I told you, there are biomolecules present like proteins, DNA and RNA. So for protein they took proteases, for RNA they took RNases, and for DNA they took DNases. Okay, so what happens is proteases and RNases did not affect transformation. Okay, but the transforming substance was not a protein or RNA. Okay, so proteases and RNases did not affect the transformation, means they were not able to stop the transformation. It means it was confirmed that proteins and RNA are not responsible for transforming R into S strain bacteria. Okay, now what they did is they use DNAs. Now that DNAs actually inhibited transformation. Why? Because DNA is the genetic material, it is getting passed from the uh, you can say S strain to the R strain, right? Now if DNA will get impacted upon by the DNAs, it means DNAs will digest the DNA, right? So if DNA will be digested, so there will be no conversion. So then they were able to confirm that yes, DNA is the ultimate genetic material which was actually getting transferred from the S strain to the R strain, so that the R strain also converted into S strain and finally killed the mice. Okay, now here I do have a question which I saw into the NCERT, and many students may get confused with that. You have two words. First is DNAs and second is again DNAs. Okay, so what do you understand by this? This is nothing but the multiple DNAs, but this is an enzyme which digests the DNA. Okay, so just remember that whenever it is being written this, it means there is a multiple DNA copies. Wherever this is the enzyme digesting or degrading, you may say, DNA. Okay, then the next what we have is, so at last what line is being written is, they concluded that DNA is the hereditary material, but not all biologists were convinced even that. Even at that time when they performed everything, they experimented everything, even all the scientists were not convinced. So what other experiments were done? Okay, first of all, moving to the other slide, I would like to tell you about more, that is Our course include, now whatever course we have prepared for you all, in that course we have 100+ classes for you. Okay, for example, you would agree with me again that in biology we have 38 chapters. Now it's not a very small task to cover all the 38 chapters, and you cannot even cover a one chapter in one class, because every chapter has their important topics, important significance. So at least it will take 3 or 4 classes to complete one chapter. So that's why we have organized, or we will organize 100+ classes for you all, so that we can cover the whole syllabus before your NEET exam, right? Second thing, we have a convenient educational platform. That's what I explained you in the beginning of the session as well, that we organize our classes, all the webinars over our YouTube channel only. Okay, once you will visit our website, you will also see over there that we have posted our YouTube links. So you can just click on those links and you will be redirected to the YouTube itself. So you can easily access YouTube, right, for our online webinars. Now we have a syllabus which is being prepared from JEE and NEET experts, right? Okay, for in JEE you have maths and additional subject. In NEET you have biology. So we have the faculties for all of the all the subjects. All of us are professional in our fields, and we prepare, we try to cover even the additional information into a PDF material for you all. Okay, so you will all be covered, you don't have to worry. Okay, then last thing, we have doubt solving sessions. Like I told, once the syllabus will be completed, you all will get a chance to be in touch with the faculties over there, so that you can get your doubts solved. Okay, and what and which is who is the medium in order to solve all doubts? You have to be in touch with your mentors first. So the genetic material is DNA now. Okay, so it was confirmed, but not all scientists were convinced. So the unequivocal proof that DNA is a genetic material came from the experiments of Alfred Hershey and Martha Chase. Just remember these names, guys. These names are very much important, okay, for your NEET exam, because many questions come on the from this topic. Alfred Hershey and Martha Chase were the two scientists who actually gave a confirmatory experiment, who actually did a confirmatory experiment to prove that DNA is the ultimate genetic material. So what they did is they worked with viruses that infect bacteria. Okay, now those viruses which infect bacteria are called bacteriophage. Okay, because virus have a very much tendency to infect anybody, any host. So those virus which have infect bacteria are called bacteriophage. Okay, then moving on, the bacteriophage attaches to the bacteria. Yes, the bacteriophage, since it's a virus, it has to get into the body of the bacteria, or on the surface of the bacteria. So bacteriophage is attaches to the body or the surface, and then the genetic material, which is the DNA or even the RNA in the case of virus, it acts as a genetic material and it enters the bacterial cell. Then the bacterial cell starts treating that genetic material as its own as its own DNA, as its own gene. So basically the DNA or the RNA of the virus is that kind of virus, that kind of genetic material, which is actually not recognized even by the cell of the host, and the host cells start treating that DNA as their own DNA. And so that they also start making copies. And once you start making copies, yes, there will be a time that the whole DNA machinery, the whole genetic the whole genome will be covered by the you can say by the DNA or the RNA of the infect the vector cell. Okay, that is a virus. So similarly, they infected a bacteria with a bacteriophage, and bacteriophage was able to take over the genetic machinery of the bacteria. Then Hershey and Chase worked to discover whether it was protein or DNA. Again, what they worked on is either the genetic material is protein or DNA. So what they did is basically they grew some virus on a medium. Now they start growing some virus in a medium which contained radioactive phosphorus. Okay, and some viruses which contain radioactive sulfur. Now you all would understand that if it's a radioactive phosphorus, and in DNA what we have is phosphate group, right? So DNA will be recognized by radioactive phosphorus, and sulfur and RNA will be recognized by proteins will be recognized by radioactive sulfur. So viruses which were grown in the presence of radioactive phosphorus contained radioactive DNA, because DNA contains phosphates. And those viruses which were grown in the culture of radioactive sulfur were found to have radioactive protein in them, but not radioactive DNA, because DNA does not contain sulfur. I hope you understand, right? What I'm trying to say is they took two things. First is radioactive phosphorus, and second what they took is radioactive sulfur. Okay, now if you would just tell me one thing, in DNA we found three things, that is a base pair, a sugar, and a phosphate group. So radioactive phosphorus will have radioactive DNA, right? Because DNA has a presence of phosphate group in them. And radioactive sulfur will have a presence of radioactive proteins. Okay, so this is what Hershey and Martha did. They took this and they were able to found radioactive DNA and radioactive protein accordingly. Then what they did is the radioactive phages were allowed to attach to E. coli. Now those viruses were were tend to attach to the surface or the body of the E. coli bacteria, and the infection proceeded. Like I said, they started to take over the machinery of that bacteria itself, that is the E. coli. Now the viral coats were removed from the bacteria by agitating them in a blender. Okay, with the help of the process of centrifugation, we can remove these viral coats. We can remove the biomolecules. Okay, and then the virus particles were separated from the bacteria also by spinning them into a centrifuge. Centrifuge is a machine in which centrifugation process occurs. Then we have now those bacteria which were infected with virus had a radioactive DNA, were radioactive. Okay. Now that virus which had a radioactive DNA, now you can imagine if the virus will attack the E. coli, so E. coli will also become radioactive. Okay, because DNA is the material, that's what you know, but they did not know. How did they concluded it? That when they checked the E. coli, so E. coli was also found to have radioactive DNA. It means that the DNA was transferred from the virus to the E. coli. Whereas when they tried to find the same thing with the E. coli, with the you can say the radioactive sulfur present in the virus, what they found is that that radioactive sulfur was not present in the E. coli. It means it was confirmed now that radioactive sulfur is not, radioactive sulfur or radioactive protein is not the biomolecule which is actually getting transferred, but what is being transferred is radioactive DNA. Okay, so that's why DNA was considered as the final or the ultimate genetic material. Even these days we have two, DNA and RNA. Okay, whereas guys, let me tell you one thing, RNA, most of the time RNA works as a messenger or an adapter, whereas DNA only works as a genetic material. Okay, so you can see this now. This is the whole picturization or a diagram which actually represents what the experiments were performed by Alfred and Hershey and Chase. Okay, so you can see that these are the bacteriophage. This one. Okay, if it's labeled as S, it means it's a radioactive sulfur and it's a radioactive phosphorus. Okay, now infection is occurring, it means it's a body of the E. coli and the bacteria is acting upon that. Once it's acting, it means now they are blending it. Blending means they are centrifuging in order to remove that virus from the bacteria itself. So what they found is that there was no radioactivity in this bacteria, but there was a radioactivity in this. It means it was found that the bacteria which was finally extracted from that blend, that bacteria was found to have radioactive phosphorus into their bodies. And if they have radioactive phosphorus, it means that they have radioactive what? Radioactive DNA, right? Guys, any confusion you have, please drop your messages in the chat box. I'll be happy to solve that out. Okay, so from Hershey and Chase experiment, it became an established fact that it is the DNA that acts as a genetic material. However, it subsequently became clear that in some virus, RNA is also a genetic material. That's what I told you. in many viruses, we can also find that the RNA is the genetic machinery. For example, when we talk about TMV, TMV is what? Tobacco mosaic viruses. Or we talk about QB, or we talk about QB bacteriophage. Okay. So these are type of viruses in which we have RNA as the final genetic material. Now we have a molecule that can act as a genetic material must fulfill the following criteria. Okay. Now, every molecule which has been found, be it in DNA or be it in RNA, they have to fulfill some criteria. What is that? Is they first of all should be able to replicate themselves, because you would understand that for a growth of an organism, replication is much needed. Without the replication, the organism, the growth cannot proceed further. The organism is not able to reproduce. The organism is not able to give their genetic information to their children, to their progeny, right? So in order to do that, the replication is much needed. So this is the very important criteria for a genetic material to have. Then material should be stable chemically and structurally. It means that it should not be highly reactive. Can you imagine, like, if your DNA is highly reactive with any drug or any chemical, then your DNA will keep on changing, and once your DNA will change, it means the whole information in your body will change. You can have mutations, okay? And once you have mutation, you don't know what's going to be happen to you. Either it will be bad or good. Well, bad in majority of the cases, so that's why DNA is very stable. DNA does not react chemically with the chemicals or with the substances. Okay, then it should provide the scope for slow changes, that is mutation. There should be some slow changes. Slow changes means evolution should be over here, like over hundreds of years. Yes, you can expect a change. Like for example, in many species, in many reptiles and amphibians, we are able to see that they have been evolved over the years. Why? Because DNA or RNA also has a tendency to change themselves, but over a time, slowly change. Okay, which will not hamper your daily living. You can see the small changes, they are required for nothing but the evolution only, and that's what I told you. Then it should be able to express itself in the form of Mendelian characters. Okay, Mendelian characters are what? Again, same thing, that Mendelian characters were able to transfer from the generation 1 to the generation 2. So similarly, DNA or RNA should also have a capacity that they can be transferred from the parent to their offspring. Okay, because of rule of base pairing and complementarity, both the nucleic acid have the ability to direct their duplication. Since DNA can also be double stranded, RNA can also be double stranded, it means they will have base pairing in them, like I told you earlier as well. Like I took a class like yesterday. So what I told you, in DNA we have 4 types of base pair. Adenine, which will which will base pair with thymine, right? So it will be a double bond, whereas guanine will base pair with cytosine in with 3 hydrogen bonds, right? Similarly, when we talk about base pairing in RNA, so adenine will base pair with uracil. There is no thymine. Thymine is replaced by uracil in RNA. This is what we have for DNA. Whereas guanine will base pair with cytosine in that. It means cytosine is common in both. Okay, this is for your RNA. So both of them have an ability to replicate themselves. It means they can produce copies of them. Okay. Stability, if we talk about stability, then yes, DNA is highly stable, whereas RNA is less stable. Okay, RNA has a tendency to mutate itself very frequently. Okay, so you can see that this now can easily be explained in the light of DNA, that the two strands being complementary, separated by heating, come together. Okay, so basically DNA was very much stable, whereas RNA was not. Okay, now further, now the second group present at every nucleotide in RNA. Now I told you that what is the difference between deoxyribonucleic and ribonucleic acid, right? In excuse me, okay, there is a pentose sugar. Okay, now in that pentose sugar, when we talk about DNA, so we do have at the second position, we have a lack of OH group over here, whereas this OH group is present at the second position in RNA. Now this OH group is highly reactive. Absolutely, there is oxygen, there is hydrogen. Either there can be dehydrogenation or there can be hydration. Hydration means formation of a water molecule, right? So you can see that since this OH group is free to react, it is highly reactive. What I'm talking about is RNA, because RNA has a presence of OH group in that, right? So that's why, being it a highly reactive, so RNA can easily be degraded. RNA is not liable to be a genetic material in many organisms. Also, RNA is known to be catalytic. It means RNA can fasten up a reaction going on into your body. Hence reactive. Therefore, DNA chemically is less reactive and structurally more stable. DNA has a good structure. DNA does not have any free molecule which is highly reactive to react with any substance. So DNA as compared to RNA is very stable and RNA is less stable. Okay. Also, RNA structure can be degraded if this molecule reacts with any other molecule present, whereas DNA structure is highly stable. Okay. Now, in fact, the presence of thymine at the place of uracil also confirms additional stability. Now, in DNA we have thymine, in RNA we have uracil. So, in thymine, thymine actually prefers, thymine gives more stability to the DNA molecule as compared to uracil in RNA molecule. Okay. Now both the DNA and RNA, they are able to mutate. Yes, I told you that you can expect a slow change in DNA as well, but high change in RNA. So RNA has a more tendency to mutate itself rapidly. Like you can see, in fact, RNA being unstable mutate as a faster rate, since it is an unstable compound and it's highly reactive, so it can mutate at a faster rate, whereas whereas DNA is very less highly stable and very less reactive, so it can mutate, but the chances are very, very, very less. Okay. Similarly, viruses, that's why we say that the majority of the viruses, they have RNA genomes in them. You would have also seen at present times the COVID-19 was firstly introduced. Then after COVID-19 we have Omicron, we have Delta, all are the members of the COVID family only, right? It means the COVID virus has an RNA genome. That RNA genome keeps on mutating itself, keeps on changing the structure and the function, and it becomes a new virus. Okay. So that's why RNA being highly mutative, it is present into the virus, and virus, that's why are called that they can mutate at a faster rate. Okay. They have a shorter lifespan, but they mutate and evolve faster. So I've given you the best example for COVID members, so you can relate to that. Okay, next what we have is RNA can directly code for the synthesis of proteins, hence can easily express the characters. DNA, however, is dependent on RNA. Okay. So now RNA is free. Okay. RNA is not dependent on anything. So if we consider RNA only, if we take RNA into the account, so RNA can easily help in the production of proteins through the process of, who will tell me the name of the process? The name of the process in which RNA converts into proteins is called translation. And which RNA we are talking about? We are talking about messenger RNA. Okay. Now you can see, but DNA cannot directly be converted into proteins. DNA first need to be converted into RNA, and then RNA will get converted into proteins. So that is what I told you about. You can say central dogma of biology. I told you this, right, yesterday. Central dogma of molecular biology, in which what I had told to you is DNA will undergo transcription. Okay, this process will be called as transcription and will get converted into mRNA. Then mRNA will undergo process of translation and will form proteins. Okay. So this is the central dogma. Okay. And this was okay. So Similarly, this is what is being written over here, that DNA alone cannot result into the formation of proteins, whereas it has to be converted into RNA first. Okay. Then what we have is both RNA and DNA, they can function as genetic material. Yes, RNA also has a capacity to transfer the genetic information from parent to offspring, whereas DNA also has that. But DNA being more stable is preferred for storage of genetic information. But how genetic information will be stored? How chromosomes will be stored? They will be stored in DNA itself. Okay? They will be stored or they will get transferred with DNA. But DNA has a property to store the information. For the transmission of this genetic information, RNA is better. DNA is better for storage. RNA is better for transfer. Okay? Because RNA also acts as a catalyst. And if in a laboratory you need to transfer a genetic molecule fastly, so you can use RNA as a catalytic molecule. Okay. Then it was found that RNA was the first genetic material. There is now enough evidence to suggest that essential life processes, whatever life processes we have metabolism, which include catabolism and anabolism in every sort. Okay, metabolism means that energy is getting used and same energy is getting produced. Okay, so that is called your metabolism. Translation, again the formation of proteins work in different kind of groups. Okay, then we have splicing, evolved around RNA. Okay, so these are all the functions which are evolving or which are going hand in hand with RNA. RNA used to act as a genetic material as well as a catalyst. Okay, we have discussed this thing 3 to 4 times into this session only. There are some important biochemical reactions in living system that are catalyzed by RNA catalyst and not by protein enzymes. Okay, now we have several biochemical reactions in which we take RNA as an enzyme but not proteins, because you can say all the hormones, all the enzymes, they are proteinaceous in nature, but we can also take RNA as a catalyst sometimes. We do not need enzymes all the time. Okay, because RNA is also catalyst and highly reactive and unstable. Therefore, DNA has evolved from RNA. So RNA basically was evolved into DNA over a period of time. It means that the first molecule which would have come is, who will give me the answer for this? The first molecule which was introduced is RNA itself, because RNA was the first. That's why it is being written that RNA was evolved into DNA, because RNA is very unstable and highly reactive. But in order to store the genetic information, it was much needed that it mutates less. How it was possible? When DNA was formed. Okay. So there was some chemical modifications which occur into the body, and then RNA formed DNA. Now DNA is being double stranded and having complementary strand further resist changes by evolving a process of repair. It means DNA, if there is a one strand of the DNA, we already have the complementary strand for the same, right? It means that we do not, it means that DNA is less reactive. Okay, because it has base pairing also. It means both the strands, they tend to be together. This strand will not react with any other molecule, this strand will not react with any other molecule. So as a result, DNA will be highly stable, DNA will be less reactive. Okay, you can see this. Now let's talk about DNA replication, guys. So it's very interesting topic. You will come to know how the copies of DNA are formed. Okay, many of you would also have studied it earlier. Let's study in detail. So now there are two strands, but those two strands, they have to be separated in order to make copies. For example, in DNA we have double strand. Okay, in DNA we have double strand. So this strand will create its own copy, this strand will create its own copy. So one DNA will produce, you can say, two daughter DNAs. Okay, so this is the process. You can say the DNA replication. Replication is what? Copy, daughter, similar words. Okay, after the completion of replication, each DNA molecule would have one parental and one newly synthesized strand, right? So for example, this was the original DNA, this was the parent DNA, Now the strands are separated. This strand is separated and this strand is separated. Now they will combine with the other strands of the other DNA. So like this, and they will form the daughter DNA, right? So what the statement says is one strand is come from the parent DNA, whereas the other strand is is a newly synthesized strand. Newly synthesized strand means that the DNA replication occurred, there was a new base pairing happened, and the new DNA, the daughter DNA, is finally produced. This is what we call as semi-conservative nature of the DNA. Just remember this line, semi-conservative DNA replication. The replication is always how? Semi-conservative. You can also see the diagram. In diagram, this is the parental DNA, and the strands of the parental DNA, they are getting separated. Adjacent to the separation, there is new base pairing occurring. It means that the new synthesis of the DNA is occurring, and these are what we are calling as daughter strands. So first is the parental, which you can plot as a blue color, whereas in the red or orangish color you can see the daughter strand present over there. So one DNA is leading to the formation of two daughter DNAs. Okay, this is what we call as semi-conservative nature of the DNA replication. Now, how was it experimentally proved? This is what you have stated theoretically, but scientists also needed to experimentally prove that. So how did they do it? Okay, so it was shown in Escherichia coli and subsequently in higher organisms. So first of all, they had proven the the DNA replication in E. coli, whereas they also took the higher organisms like plants and human cells. And finally, Matthew Michelson and Franklin Stahl, they together performed an experiment in 1958 into in order to prove that DNA replication is semi-conservative in nature. What they did is they grew E. coli in a medium containing ammonium chloride. NH14 is ammonium chloride. Okay, NH4Cl, sorry. Yeah. And 15. Now, if you know what is the atomic number or what is the mass number, so you can say that the mass of the nitrogen atom is 14, but where here it is 15, it means it is the heavy isotope of nitrogen. Similarly, what we have is the only nitrogen source for many generations. The result was the 15 and was the incorporated into newly synthesized DNA as well as other nitrogen containing compounds. So Michelson and Stahl, what they did is they took this heavy DNA molecule, they took ammonium chloride, they actually replicated the DNA into this ammonium chloride having a mass number 15, means heavy isotope of ammonia, heavy isotope of nitrogen. Okay, now after that, what was happened, that the DNA which was actually produced, and that DNA was consisting of this N15 heavy isotope of nitrogen. Now it was easily be able to distinguish from the normal DNA by centrifugation in cesium chloride. How were they able to distinguish which one is heavy DNA and which one is the normal DNA? They were able to basically centrifuge that in which you can say chemical cesium chloride. Okay, and because of the density gradient, because you always know, you always are able to study in physics also. Okay, Nihal is asking कब से start की NEET classes. Actually, Nihal, this class was started at 3:30. Okay, so you can also view the playback of this video if you have missed anything. Okay, so what happens is, now after the centrifugation which was happened by Michelson and Stahl, what they did is they were able to identify which one is heavy and which one is normal, because heavy DNA always tend to move, always sediment downwards, right? You know this rule, right? That the heavy part sediments whereas the light part flows. I also explained the same thing when I was explaining gel electrophoresis in order to fragmentize the DNA parts, right? So now what they did is, then that they transferred the cells into a medium with normal ammonium chloride solution. Now what they did is, earlier they basically transferred the DNA into a heavy isotope of nitrogen. Now they took the normal isotope, which is N14, uh, ammonium chloride with a 14 isotope of nitrogen, and then they took the samples after a definite time interval. Then the cells multiplied, that is the DNA was multiplied or replicated. Now they extracted the DNA, and what they found is that the various samples were separated, and now they measured the densities. So what they found is that you can see in here the DNA that was extracted from the culture 1 generation after the transfer from 15 to 14 medium. Now what they did is they they tried to combine the DNA strands which were obtained from the ammonium chloride with having a nitrogen isotope 15 with that DNA which was having a nitrogen isotope 14. So you better are able to relate the genetics concept in this chapter. How? That, for example, this is the N15 DNA. Okay, let me just write it over here. N15 DNA. Both the strands are N15. Now what they did is they combined this with N14. So they found that there were two DNAs produced, in which one strand was N15 and the other strand was N14. Okay, similarly over here they found the other daughter DNA in which one strand was N15 and the other strand was N14. Now what they did is they again replicated these daughter DNAs, and what they found is now there will be 4 copies of the DNA because one will lead to 2 and 2 will lead to 4, right? So they found is N14 DNA, pure N14 DNA. Again, one strand was N14, the other strand was N15. One strand was N14, other was N15, and lastly N14 DNA. Let me show you the diagram, what I am trying to explain. So this is what is the heavy isotope, the heavy DNA, which is consisting of N15 nitrogen DNA, right? Now what is being done is it is after 20 minutes the DNA replication is occurring into the E. coli bacteria. E. coli bacteria have a tendency in which DNA replication is totally processed after 15 minutes only. So the first strand was N14, the other is N15. Similarly, in generation 2, when the replication proceeded, so N14 DNA were produced, two daughter N14 DNAs, whereas one was again N15 N14 and N15 N14, two strands. Okay. I hope this concept is clear. This is a separation of DNA by centrifugation, and this experiment was done by Michelson and Stahl. Okay. Now the machinery or the enzymes. Now we'll study about what are the enzymes which are responsible to carry out the DNA replication. So enzymes are like in E. coli. Basically enzymes are nothing but the catalyst only. Okay, catalyst enzyme. So the main enzyme which is referred to as DNA dependent DNA polymerase. Okay, so which is the main enzyme which carry out the DNA replication processes? DNA dependent DNA polymerase. Okay, since why it is called DNA dependent? Because DNA polymerase catalyst or enzyme uses the DNA template in order to carry out DNA replication. Okay, that is. Now what is replication? Replication is nothing but polymerization of the nucleotides. More nucleotides are adding, it means new chain of DNA is getting produced. Okay. These enzymes are highly efficient enzymes as they have to catalyze polymerization of large number of nucleotides in a very short time. Why do we need catalyst? In order to fasten up a reaction, and we have to get the DNA replicated in a very short period of time. So that's why we need to add the catalyst. We are not adding these catalysts, but these catalysts are present naturally and they are carrying about the DNA replication into our bodies. Similarly, not only do these polymerases have to be fast, but also these polymerases catalyze the reaction with high degree of accuracy. You can not only expect the faster rate, but you also can expect accuracy with that. If there will be no accuracy, then you can also imagine that there will be mutations, there will be a bad impact on the person, physical appearance, mental appearance, anything, any single thing can happen if there will be a change in DNA. So it has to be very accurate when it comes to replication, as well as it has to be faster. So polymerase fasten up the process, whereas it maintains the accuracy also. Okay. So furthermore, energetically replication is a very expensive process. Expensive, it means that it doesn't mean that you have to expend money. No, expensive means it means it needs very high amount of energy in order to carry out replication. So deoxyribonucleoside triphosphates, they serve dual purposes. Now these are the molecules which we call as deoxyribonucleoside triphosphates. What they do is they first of all act as substrates which provide energy for polymerization, that is the DNA replication, whereas and those are the two terminal phosphates in a deoxynucleoside triphosphate are high energy phosphates. Okay, now since the name suggests deoxynucleoside triphosphate, it means there are three phosphate molecules. Out of these three phosphate molecules, two phosphate molecules are responsible to provide high yielding energy. Okay, same as that the ATP provides. Okay, so where does the energy come from? Deoxynucleoside triphosphate. Just remember this term as well. This can come into the form of an MCQ for you all. Next we have, in addition to DNA dependent DNA polymerase, there are other enzymes also which complete the process of DNA replication for you all with high degree of accuracy. What are those? For long DNA molecules, since the two strands of DNA cannot be separated in the entire length, this does not mean that, for example, if this is a strand, this is a whole DNA molecule, then first of all it is not necessary or it does not happen that DNA will separate like this. No, if this will be separate like this, then where will the genetic information stored? So what does it happen? Only at a particular site the separation of the strands occur, either this, either that, any other side. Okay, it means a small portion of the DNA starts separating, like regularly. So the replication occur with a small opening of the DNA helix, referred to as replication fork. Now whatever part of the DNA where the replication is, where there is a small opening there, where there the DNA replication actually starts from, that point is called as replication fork. How do you see a fork? Fork is like in this shape. It means if this was the molecule, so these are the two strands which are separated from one another. That's why this portion will be called as replication fork. Okay, next what we have is the DNA dependent DNA polymerase catalyze polymerization only in one direction. Absolutely, they have to catalyze the reaction only in one direction. Now the direction will be 5 to 3 direction. It means the fifth and the third end are always free, so the reaction will proceed from the fifth end and end will with the third end. This creates some additional complications at the replicating fork. What are the complications? We'll study. Consequently, on one strand the DNA, the template with polarity 3 to 5, the replication is continuous, while on the other template with polarity 5 to 3, it is discontinuous. Now you can imagine, even I told you that in DNA, for example, these are the two strands. So one strand will have 5 to 3 polarity, where the other will have 3 to 5 polarity. Simply, when there is a DNA, okay, like for example this, and this is called the fork. So if this is 5 to 3 polarity and this is 3 to 5 polarity, so as I told you, replication always occur in 3 to 5 polarity, 5, right, 5 to 3 polarity. It means that the strand which has an opposite polarity will easily be replicated. This strand which has 3 to 5 polarity can easily be replicated, but since the replication occurs in 5 to 3 only, whereas the polarity of the of the strand is the same, so it has to be you can say replicated in small groups, small fragments. Okay, and those fragments, they first have to be converted into fragments, so then the application occurs, and after that those fragments have to be joined together by which enzyme? DNA ligase. So just remember this, whenever two fragments have to be joined together, that process is carried upon by DNA ligase. Okay, so this is the replication process. DNA polymerase initiate, DNA polymerase bring about replication, and DNA ligase join the fragmented portions. So Now I told you like there is a replication fork, okay, which actually resembles to the separation of the DNA fragments. Also there is an origin of replication in the DNA molecule. That origin of replication represents that from that point only the DNA replication will start, okay. So you need to remember this, the point from which the DNA replication starts is called origin of replication, okay. Why it is needed? Because recombinant DNA procedures require a vector, and vector can only recognize origin of replication. Vector will be there, it will attach to a DNA molecule, it will attach to the origin of replication, and from there DNA polymerase will bring upon the DNA replication. The replication fork will occur, and replication fork will be formed, and DNA polymerase will start functioning. So now this DNA replication in eukaryotes is happened in the S cycle of the cell cycle, S phase, okay. Yeah, just remember this thing, S phase के अंदर DNA replication होती है when we are talking about eukaryotic organisms, okay. Also, now cell division and DNA replication, they have to be correlated, they have to be cooperated with each other. If there will be DNA replication is happening but cell division is not happening, it means there will be polyploidy. Polyploidy means the chromosomes will not distributed to the daughter cells because there is no division, but the chromosomes are you can say the chromosomes are producing. It means more chromosomes means more genetic information, that is polyploidy. Polyploidy is also not good for an organism, okay. So cell division and replication has to go and hand in hand. You can see this is DNA strand 5 to 3 polarity and 3 to 5 polarity on the other hand. So you can see in the 5 to 3 polarity strand, the replication is discontinuous, whereas in 3 to 5 polarity strand, the replication is continuous, okay. This is what I tried to explain to you guys. So guys, this was all for today, okay. I hope that I was able to make you guys understand about DNA replication and how DNA strand was finally concluded that yes, it's a genetic material. Okay. So replication is a very interesting part. You came to know about DNA polymerase. You came to know about the energy currency, which is deoxynucleotide triphosphates. You also came to know about DNA ligase, replication fork, origin of replication, why origin of replication is needed so that the vector can join through that origin of replication, okay. So this was all for today. I hope you will be able to learn something from this session. Every concept will be will made clear to you guys. So this is the QR code of the Telegram channel. Again, you can just join scanning through this QR code. Thank you so much guys, this was all for today. My name is Pratyaksh and I wish you best of luck. Thank you.